Today we are going to discuss about the bacterial cell wall. In the previous lecture we have done about the lecture about the bacteria structure. Now we are going to discuss about the bacterial cell wall. Basically, bacterial cell wall is composed of murine. Now what is murine? Murine is basically a mixture of two conjugated molecules. Now which are the two conjugated molecules? First is peptidoglycan and second is lipoglycan. We'll be discussing about the structure of peptidoglycan, but the lipoglycan is basically, it is present, that is, they both are the conjugated molecule, and lipoglycan is basically present in gram-negative bacteria. We'll be discussing what is gram positive, what is gram negative. Now, cell wall is basically classified on the basis of its composition. The bacterial cell wall is basically classified on the basis of its composition. Now, there was a scientist named as Christian Gram. He basically developed the staining technique. He developed the staining techniques, means the he stained some bacteria and he classified those staining response, he classified on these bacteria on the basis of staining response. First is there's response, he has classified on the basis of the bacteria on the basis of staining response. So gram positive bacteria and one will be gram negative bacteria. Gram positive bacteria means the staining response will be positive but on the grams staining, and here the staining response will be negative on the grams staining. Okay, so that he has classified the bacteria on the basis of the staining techniques or the staining response, that is gram positive bacteria as well as gram negative bacteria. Now if we talk about the peptidoglycan structure, its composition is basically based on the peptide chain with sugar, as peptide and glycan. It means peptide chain with the sugar. Its composition is basically based on these. And which type of sugar? One is N-acetyl muramic acid and one is N-acetyl glucosamine. Okay, these are the sugars of the peptidoglycan, peptide chain with the sugar, and these are the sugars. Now what is N-acetyl glucosamine? N-acetyl glucosamine. Basically it includes acetyl plus glucose plus amino group. When they combine together, they make N-acetyl glucosamine. When N-acetyl glucosamine will combine with the lactic acid, they will form a N-acetyl muramic acid. Okay, what is N-acetyl glucosamine? When acetyl glucose amino group, these three will combine together, that will make N-acetyl glucosamine. And when N-acetyl glucosamine will combine with lactic acid, it will make N-acetyl muramic acid. And the peptidoglycan is basically a chain of the N-acetyl muramic acid as well as N-acetyl glucosamine. Now let's see the structure of the peptidoglycan. This is the structure of peptidoglycan. When like N-acetyl muramic acid is linked with N-acetyl glucosamine, then N-acetyl muramic acid, then it is linked with the N-acetyl glucosamine, then N-acetyl muramic acid. Same here, they both are linked together, and these are basically the short tetrapeptide. And what is short tetrapeptide? That is basically made up of, this chain is basically made up of alanine, glutamic acid, lysine, and then alanine. So these four are, and when like these two, when the here in mid between glycine is present, when glycine is joining, when glycine join two short tetrapeptides, that is known as pentaglycine linkage. Like when these are the short tetrapeptide, and when glycine is present mid between them, when glycine attach with the two short tetrapeptide chain and they make a complete chain, that is known as pentaglycine linkage. We can say that the bacterial cell wall is basically composed of peptidoglycan as well as lipoglycan, because as I said, it is made up of murine, so it means that it is made up of peptidoglycan as well as lipoglycan, and that is the basically structure of peptidoglycan.